Hello and good morning. I did it again. I recorded myself without the slides, so I'm going to re-record it, and hopefully you'll get the right outcome. Okay, continuing on with the genetic code. Right, so this slide, just again, I've mentioned this in the first slide, I think, when we talked about genetic code. So it's not ambiguous, right? So it's very direct. So I think it's clear from the smallest of organisms like the bacteria to the largest of animals. They use the same code as well in plants as well. There are some ambiguity, some exceptions. In vitro, that means when you start cloning, sometimes UUU, which is supposed to code for phenylalanine, you get leucine sometimes, right? So it's confused, but it doesn't happen in vivo. In vivo, inside the cells, in real life. In real life, it's not confusing, it's not ambiguous. You only see ambiguity when you talk about experimental system. We've also gone through this. Once you start reading, you just continue, and it doesn't read in an overlapping. This is not how it's being read. There's no comma, there's no doesn't stop, and what else? All right, no gaps. Okay, so basically an mRNA, if this is an mRNA, right, you'll have, remember the coding sequence is in the middle, well, somewhere in the middle of the mRNA. It doesn't start right at the beginning of mRNA, right? At the beginning, what is this sequence? This AGG. Remember that? This is the Shine-Dalgano sequence. And what's the Shine-Dalgano sequence? This is where your ribosome sticks, right? Your ribosome binds. Remember, the 16S and the mRNA will bind at the Shine-Dalgano. That's the Shine-Dalgano sequence. This is where, and that binding will cause the AUG to be placed in the P site. Remember, all right? So you have the start codon and also the stop codon. The regions that are not being translated, what are they called? They are called the untranslated region, right? So you have the 5' untranslated region, and then you have the 3' untranslated region. The coding sequence are called the ORF. ORF means the open reading frame. So any sequence that can be translated is called an ORF. Let's say you found a new bacteria, you sequence, and then you see a piece of you sequence, you get your sequence, and then you translate. You can see by referring to the codon table, you are able to translate, right? So you think it's a gene, but you cannot call it a gene yet, because you haven't really done experimental, you don't have any experimental evidence. But because it can be translated, so and it has a frame of reading frame, so you call it, then this is what you call as the open reading frame. All right, okay, then we will go into mutations. It's easier if you see the examples later, but this slide talks to you about deletion. This is the middle sequence. This is the original sequence. You start with AUG, AAA, CCC, GGG. All right, but because there is, okay, an example here is there's a deletion. You remove the A, because the nature of reading of genome sequence is non-overlapping, it's continuous. So that means it's going to stay, your AUG is still correct, but then it's going to start AAC, CCG. See how it's different from the original? Because you remove. It's the same thing if you add a sequence, right? You add a base. So now you say it's AAA, fine, it's the same one. Then suddenly you have GCC, right? Originally CCC, but because it has moved one base, because the addition of one, So now the frame, the reading frame, remember open reading frame, the 3x3. So now the original reading frame has changed. Sometimes it's called, change is also called a shift. So this is why addition or removal of how many bases, doesn't matter. If there's any insertion or deletion, addition or removal, it will actually cause a frame shift. So this is one mutation that causes the reading to be. We'll show you what it means later. This is the examples to show that the codons. For example, here I mentioned the UGA, a normal situation is a stop codon, but in the human mitochondria is a tryptophan. AUA, for example, normally it codes for isoleucine here. In the human mitochondria, it actually codes methionine. So it's nearly universal, but there are some exceptions. This is collinearity from the gene to transcription to mRNA, mRNA to amino acids. There's collinearity there. So now let's talk about mutations. You know what's a mutation? It's a change in amino acids. It's a change from the normal. If you translated the whole book, or if you photocopy the book, and suddenly when you check again, one whole page of text is missing, so that's a mutation. That's a deletion, right? So anything that's different from the original is called mutation, right? It gives you mutant. So I have this question in your exams, in your test or your quiz, and quite a lot of you actually got it wrong. I think I mentioned in class, I think you weren't paying attention. A point mutation is a small change. Previously, when I was studying, a point mutation says a change in one base, but nowadays, as I say, as many as 5 bases, if there's a change in 5 bases, it's also called a point mutation. Right, so I know 1 or 2, 3, 4, as long as within, at least 5 bases are changed, this is called a point mutation. All right. I think because this one gives you a single, it says there's a single base pair. Right, but never mind. I'll probably give you some free marks in the quiz. So a transversion and a transition. A transition is a change from purine to purine, pyrimidine to pyrimidine, right, within the same class. But a transversion is a mutation from one to the other. So a transition is in blue. So A to G, right? So that means A is a purine to purine, G to A, C to T, or T to C. These are all transitions. But when A changes to a C or A changes to a T, these are transversions. So what I'm talking about. How many transitions are there? Possible transitions, there are 4 possible transitions, and then there are 8 possible transversions. Here you go. So I think this is where you got confused. It says a change in a single base pair, but later in the slide it says point mutation, sometimes up to 5 base pair. Right? So it's still point mutation. So it can either be substitution, a change, it can be either deletion, or it can also be an insertion or addition. Okay? Now, mutagenesis, we're going to look at what causes mutations. Alright? I'll see you in the next video.